All right. Thank you, everyone, for attending the Needham Healthcare Conference. It's my pleasure to introduce Amit Munshi, President and CEO of Arena Pharmaceuticals.
Good afternoon, and thanks again for the introduction and for the invitation. Excited to be here today and excited to talk about where Arena has been over the last couple of years, but more importantly, where we're going over the next few. Before I move forward, I'd like to just remind everyone of our forward-looking statements. Our full disclosures are available on the SEC website in our SEC filings. So Etrasimod, for those of you in Arena, for those of you that have not been following the story, is a bit of a transformative play over the last 3 years. We came in as a management team in 2016 under the premise that there were some incredible compounds developed by the Arena historical discovery research engine that were worth spending time, energy, attention on. We inherited the company around a $400 million market cap, and today we sit at about $2.5 billion market cap with about $1.3 billion in cash on our balance sheet. The focus of our efforts over the next couple of years are going to be to move Etrasimod, our lead program, forward in ulcerative colitis and Crohn's disease, and we'll be starting phase 3 trials imminently on both of those programs. We're also taking Etrasimod into additional indications, and we're starting with atopic dermatitis. That will initiate this year, following with Olorinab, our second product in IBS pain, also initiating this year. A couple of these programs will read out in 2021, followed by additional readouts in 22 and 23. So we have a really exciting next couple of years for the company. We believe that the last 3 years are just the beginning for the company, and we think the best is yet ahead. So let me start by talking a little bit about Etrasimod, bringing folks up to speed on where we're at with the program. We believe this is a next generation, a potential best-in-class S1P receptor modulator. This was developed in-house at Arena through a decade of work on GPCR biology and discovery research. And this compound, along with all the other compounds that I'll describe for you in the Arena portfolio, are really designed from the bottom up. And there's some common themes to all these compounds. They have best-in-class pharmacology, they have best-in-class pharmacokinetics, pharmacodynamics, and we'll share a little bit of that information here today. So Etrasimod, as I mentioned, we believe is a best-in-class receptor modulator against S1P1 with activity on the 4 and 5 receptor. It does not have activity on S1P2 and 3. Most of the other agents out there have demonstrated activity against S1P2, which has some deleterious effects, and S1P3. The drug has a very rapid onset and offset of action. The drug has a short half-life, about 36 hours. It has no major metabolites and has an extremely safe cardiovascular profile. The drug does not require titration to blunt any cardiovascular effects. And with this profound safety profile that we've seen to date, we've also been able to deliver on best-in-class remission. And we'll talk a little bit about that. We think Etrasimod across UC, Crohn's, atopic derm, and other indications we'll proceed with over time represents somewhere between a $4 and $8 billion market opportunity. We'll talk a little bit about the market before we dive into the product. One of the questions that comes up quite frequently is, what is the market opportunity for a novel oral product in ulcerative colitis and Crohn's disease? There's a lot of companies working on lots of different modalities in this space. I think the first and most important thing to remember that despite biologics being on the market for close to 2 decades in UC and Crohn's, up to 80% of patients with moderate to severe disease are either not on a biologic, never received a biologic, or are failing their biologic treatment. Let me just reiterate that. 80% of the patients out there, moderate to severe disease, have never received a biologic or are failing the current biologic. So we think the market opportunity here is quite substantial. Several market studies have suggested this market could be upwards of $30 billion by 2026. The preference is moving to orals. And despite new biologics being in development and biosimilars on the horizon, more and more physicians are representing in in market analysis that they will be moving more patients to orals earlier in treatment. There are two primary classes of oral agents in development for ulcerative colitis, the S1P modulators, of which, again, we believe etrasimod is the best-in-class agent, and the JAK inhibitors. And there's multiple JAK inhibitors in development. Physicians routinely select the profile of S1P modulators over JAKs simply because S1P modulation is far more specific. We work on very specific types of immune cell types, and we retain immunosurveillance. All the various issues with the JAKs, including malignancies, increased infection risk, herpes zoster, tuberculosis reactivation, and of course the thromboembolic events are not seen with the S1P modulators. And looking at the two main next generation S1P modulators, etrasimod and ozanimod from Celgene, now BMS, etrasimod is selected 8 out of 10 times by clinicians. So we're really excited about the profile of the compound, and the market data continues to suggest that we're in the right space at the right time with the right product. One of the bigger issues that's arisen over the last couple of months is this risk of thromboembolic events with the JAK inhibitors. And I think it's important here just to take a moment to understand that the IBD population is different from the rheumatoid arthritis population, as an example. The rate of VTEs in the general population is about 1.38 per 1,000 person years. The rate in RA, for example, is slightly elevated to 1.7, but the rate in IBD is double. So patients with IBD have a 2x inherent risk of thromboembolic events. Patients who are treated with JAK inhibitors show about 11 cases per 1,000 person years. So the question becomes, would you select a JAK inhibitor, or would you select an S1P modulator for patients with IBD? And this analysis, along with everything we've seen so far and the overall safety profile, continues to warrant that S1P may become the preferred class in IBD. As I mentioned, etrasimod offers better control than ozanimod. We have a faster onset of action, as shown on the right-hand panel, about a 3 times faster onset of action, and we have a much faster offset of action. At 2 weeks with ozanimod, only about 60% of lymphocyte recovery has occurred. With etrasimod, within a week, we have 95% recovery. So you can move the lymphocytes down faster, and you can recover them faster. It offers the potential for physicians to have greater control over therapy. We previously discussed our 4-domain data from our 12-week phase 2 OASIS study. And just to refresh everyone's memory, we showed an 18.7 delta over placebo. A little over 24% of patients had remission as defined by the 4-domain rating scale. And seen on the left, that's roughly twice the magnitude of effect seen with ozanimod. This is despite the fact that we had a far more recalcitrant patient population. In 2016, the FDA changed the definition of remission for clinical development to a 3-domain remission scale with a far more stringent requirement of rectal bleeding being scored at 0. On that definition, etrasimod looks even better. So, and with that definition, 31% of patients hit remission at the 12-week endpoint. Importantly, we reported out late last year critical data from our open label extension study. For patients who were re-randomized into the open label arm, 39% achieved 3-domain remission. And patients who had started on 2 milligram and stayed all the way through to the end of the approximately 1-year open label treatment period, 50% had 3-domain remission. So that data continues to get better as patients go out in time. And just to give you a point of context as to what the sustained efficacy comparisons look like relative to other agents. Agents like vedolizumab, durability of clinical remission, only about 20% of patients are still in remission at the end of a year. With the JAK inhibitors like tofacitinib, you see numbers in the 30 to 40% range. In the case of etrasimod, 75% of patients of the 2 milligram group had sustained and durable clinical remission, and importantly, 93% of patients at a year had sustained durable clinical response. So these patients stay on the drug, they continue to improve, and we believe this is, again, the hallmark of a potential blockbuster product. Our safety profile continues to be consistent with what we saw in the 12-week data. There were no new safety findings in the open label extension that were inconsistent with what we've seen before. The drug is generally safe and well tolerated. Adverse events were mild or moderate. There were no SAEs at the 2 milligram group, and there were no SAEs related to any cardiac conduction issues. The impact on heart rate was predominantly approximately single digit across the study with no titration and low rates of AV conduction abnormalities. There were, again, reiterating, no SAEs related to heart rate changes or AV conduction. Importantly, unlike ozanimod, we've seen no cases of sinoatrial arrest. We've seen no increase in liver function abnormalities, macular edema, or pulmonary function testing. So we're really excited about the safety profile of the compound, and the longer we go out in time, we continue to reiterate that we think we have a compound here that has not only best-in-class efficacy, but potentially a best-in-class safety profile as well. One of the big areas of discussion at the S1P modulation is cardiac conduction and cardiac safety profile. So I just want to take a minute and compare etrasimod to ozanimod. Ozanimod has a higher rate of conduction at the GERK channel, the G protein inward rectifying potassium channel that's found on cardiac myocytes. Etrasimod has an optimized S1P profile and GERK potency. In fact, etrasimod is 10x increased potency at the S1P1 receptor with 2x less potency on the adjacent GERK channel. This is why we don't see significant heart rate drops as ozanimod did, approximately a 20 beat drop with their 1 milligram in a non-titrated schedule. They then subsequently need to go to a titration schedule to blunt that effect. Etrasimod with 2 milligrams or high dose had approximately a single-digit heart rate drop across the trial, and no titration is required. We've seen no evidence of any conduction abnormalities after day 1, whereas ozanimod's seen conduction issues out to day 8. In fact, their QT trial showed the greatest heart rate effects out to day 8. So we were seeing a better AV conduction profile, a better heart rate profile, and a better overall safety profile for the compound. We're putting the final touches on our UC phase 3 trial. This program is designed in conjunction with regulatory agencies around the world. The program is designed to start mid-year. The first study will be a 52-week maintenance study, a 52-week study that will look at patients in a 2-to-1 randomization scheme versus placebo. Half the patients will be naive to biologics, half will have previously been exposed to biologics. The second study, using the exact same inclusion criteria, will be a 12-week study, and that was conventionally known as an induction study. And patients from both studies will move into an open-label extension. Okay. One of the big exciting areas for etrasimod is the broad range of potential indications. About 2 years ago, we did an analysis of over 110,000 pieces of literature looking systematically at 23,000 autoimmune and inflammatory conditions. We identified 431 that were S1P mediated in the literature, and we further did an analysis to drive that down to about 35 indications in 7 therapeutic areas that are, we believe, have a high probability of technical success and have the ability to make a real impact in patients' lives. One of those that floated to the surface was atopic dermatitis. Atopic dermatitis has many of the same hallmarks as ulcerative colitis. You've got a disruptive barrier, again, like ulcerative colitis. You've got activation of an immune cell type with an insult, like ulcerative colitis. And then you've got activation and release of T lymphocytes to the site, again, similar to ulcerative colitis. So we believe that the mechanism here fits right on top of what we think our product does. We had some early interesting data in a variety of dermatologic conditions. Here you see a patient who presented with pyoderma gangrenosum, a very severe form of ulcerative skin disease, and saw about a 70% reduction at 12 weeks. The physicians who treated these patients were incredibly impressed, and these patients stayed on the drug for a longer period of time. So we expect to start the atopic dermatitis study this year, and we expect to have data next year from the phase 2b study. So shifting gears to Alorinab. Alorinab, our second product, is a peripherally restricted, highly selective full agonist to the cannabinoid 2 receptor. It is the only molecule of its kind, as far as we can tell, in development. It is 1,000-fold more selective for CB2 versus CB1. It is a full agonist, means it completely internalizes the G protein receptor. And we think that has applications, it can manifest itself in terms of tachyphylaxis, having partial agonism can. And importantly, the drug was designed to not cross the blood-brain barrier. So it's designed to be peripherally restricted. And this is all important because when you're tackling cannabinoid receptors, the natural inclination is to think about the opportunity for addiction or any psychotropic effects, and as we'll talk about in a moment, we haven't seen anything of that, anything associated with either one of those in our phase 2 explorations to date. The market opportunity here is quite impressive. The CB2 receptor is expressed throughout the gut. It's overexpressed in multiple gut conditions. It's overexpressed in the margins of Crohn's patients. It's overexpressed in patients with IBS. So we think we have an opportunity here to really build a gut-specific pain product. As I'll talk about in a moment, we did some initial explorations in patients with Crohn's disease who had quiescent Crohn's. So the inflammation was tamped down, but they still had durable pain. And in fact, upwards of 90% of patients with Crohn's disease and ulcerative colitis experience consistent pain independent of the management of their inflammation. And the similar number in the IBS side. So independent of the motility agents, independent of managing the motility, patients still experience significant pain. So we think this is a fantastic area to do work in. We think this has the opportunity to make a real effect in these patients' lives. Importantly, there's an interesting shared mechanism between IBS and IBD pain, and it's what we refer to as submucosal microinflammation. So you in a Crohn's setting, you have damage, you have inflammation. When that inflammation is tamped down, you have this microinflammation that continues at the submucosal level and continues to create pain for patients. As I mentioned before, the CB2 is then expressed in both IBS and IBD, both in humans and in animal models. We conducted a relatively small exploratory phase 2A trial. With all the caveats of a small trial, we were very impressed with the information. We looked at 2 doses, 25 milligrams at the low dose. We knew that 25 milligrams was sufficient to have full receptor activation, and then we tried one higher dose just to look at the tolerability of the drug. We were hoping and expecting to see somewhere around a 2-point change on a 10-point pain scale. And this is the pain scale that's used for approval with the FDA. It's the AAPS pain scale. What we saw was a mean change of 4.6, so substantially higher than products currently on the market, and at the very high end of our expectations for where this product would have worked. The FDA approval endpoint is a 30% improvement in the AAPS scale. And what percent of patients hit that 30% threshold? In our case, 100% of patients at week 8 had met that threshold. So we think we're definitely barking up the right tree. Again, with all the caveats of a small study, we think we're definitely barking up the right tree here with gut pain. We expect to start this year a study in IBS pain, a phase 2B study, and we expect to report that out next year. Okay. So I'll wrap up with talking about one of our early stage compounds. As we've discussed previously, and for those of you who've been following the story, Arena has a basket of compounds. We have additional compounds sitting on the shelf. And over time, it was our intention as our cost of capital and our capacity improved to move some of these compounds forward. The next compound up in our pipeline is APD-418. It is a first-in-class calcium-independent myofilament D repressor for acute decompensated heart failure. And we're incredibly excited about the potential of this compound. It's a preclinical stage compound. We expect to file an IND this year and be in clinic next year. So let's talk about the unmet need here in decompensated heart failure. There are close to 10 million acute heart failure hospitalizations in the United States, or projected to be about 10 million. It's roughly 9 million today. 70% are readmitted within 1 year, 20% increase in mortality with each subsequent hospitalization. There are no approved treatments to improve outcomes for these patients. The drugs which are used, the inotropes, specifically drugs like dobutamine, not only have decreased efficacy, but have a significantly higher 30-day mortality rate. So you solve the problem near term, but you increase mortality. And those are the only options on the table today. APD-418 works in a completely novel way. It hits a receptor called the beta-3 receptor. The beta-3 receptor works in the heart, is expressed on cardiac myocytes in a stress situation. So when the heart goes into failure, the beta-3 receptor is expressed, and its job is to put the brake on the heart. So it acts as a brake on the heart. Our drug works through the beta-3 receptor, taking the brake off the heart, thereby increasing contractility. But it does so in a calcium-independent manner. And doing that in a calcium-independent manner means we don't see any changes in hemodynamics for the patient. We've looked at this product in multiple animal models, and you see some of the dog data here on the right. You can see that in a dose-dependent manner, the drug improves ejection fraction and cardiac output with no change on mean aortic pressure or heart rate. So we hope this promise of this drug that we've seen in the animals will be seen in humans, and we expect to be in humans early in 2020. So just taking a step back, we've been on a pretty exciting 3-year journey. We've had 3 major phase 2 data readouts, multiple open label data readouts. We've cleaned up a lot of the historical complexities of ARENA, including obligations that we had 3-4 years ago. We've stripped out a lot of the historical costs out of the system. We've rebuilt the management team. We've reconstituted a large part of the board of directors. We built a whole new management organization, and we're in a position over the next couple of years now to deliver multiple phase 2 readouts and multiple phase 3 readouts on compounds which we continue to believe are potential best-in-class agents. So we're really excited about what's on the horizon for the next 3 years, and we appreciate all of your support and look forward to sharing more information with you later this year and then early into next year. So thank you again. Okay. Thank you.